Welcome, everyone. I am Jessica Daniel. I am with EPA's Center for Computational Toxicology and Exposure in the Office of Research and Development, and we're happy to have you with us today. As I'm going through some introductory slides, we welcome you all to say hello in the chat. If I can also remind everyone to please mute yourselves as well, that will help us as we are going through our session today. So today's training is part of EPA's new NAMS pilot training program. NAMS, or New Approach Methodologies, are defined as any technology, methodology, approach, or combination that can provide information on chemical hazard and risk assessment to avoid the use of vertebrate animal testing. EPA is working on development and application of NAMS to reduce the use of vertebrate animal testing and support more rapid chemical safety evaluation. The goal of the NAMS pilot training program is to develop, implement, and maintain an engaging set of training materials and opportunities to further understanding, confidence, and application of NAMS to chemical risk assessment. These trainings include interactive case studies to encourage active learning, train the trainer sessions, and opportunities for you, the attendees, to provide feedback on what is useful and how we can improve this program. Today's ECOTOX Knowledgebase training is the first topic featured as part of this program. Future virtual and in-person trainings are being planned on other NAMS tools and resources, which will be listed at the end of this presentation. Please visit the EPA NAMS training website listed here on this slide for additional information. So let's get into the agenda for today's training. We'll provide some welcome and introductions. We're already halfway there. We'll give you some background on ECOTOX, cover some basic features of ECOTOX, and then provide a demonstration of the knowledge base. Then you will get to walk through case studies from ECOTOX using the tool itself and really get some hands-on experience with the knowledge base. And then finally, we'll come back together and have a summary report out in learning from each of you. So now I'll introduce today's presenter, Dr. Jennifer Olker. Dr. Jennifer Olker is the coordinator for the ECOTOXicology knowledge base from EPA's Office of Research and Development, Center for Computational Toxicology and Exposure, within the Great Lakes Toxicology and Ecology Division, stationed in Duluth, Minnesota. Dr. Olker is involved with systematic review and curation of ecotoxicological data, research on endocrine disruption utilizing in vivo and in vitro assays, and development of adverse outcome pathways. Prior to joining the EPA in 2015, Dr. Olker received her PhD in Integrated Biosciences from the University of Minnesota and was at the University of Minnesota's Natural Resources Research Institute for more than 10 years, conducting research on the effects of anthropogenic stressors on aquatic ecosystems. Jennifer, the floor is yours.
Thank you, Jessica. Well, thank you everyone for being here today. I am super excited to share the ECOTOX training with this group and welcome questions throughout the presentation and demonstration. We do have a large group today, so it may take a little bit to get to your questions, but we've got folks monitoring the chat, so we will get to them as soon as we can. First, we're going to cover just a little background and some history on ECOTOX. ECOTOX started over 30 years ago, first as the Aquatic Toxicity Information Retrieval, or AQUIRE database, which initially stored only acute laboratory data. In the early 1990s, major changes were made to AQUIRE to expand the types of data that were extracted, including outdoor field exposures and studies on chronic toxicity. Not much later after that, the ECOTOX project received funding to combine 3 separate EPA databases, AQUIRE, PhytoTox, and TerraTox, to create one main searchable database for aquatic organisms, terrestrial plants, and wildlife. This was released as a web-based interface system in the late 1990s. Much more recently, we have done a complete update and expansion of the ECOTOX web-based user interface to incorporate interactive querying and data visualization options. ECOTOX was developed for and continues to support the need for an authoritative source of ecological toxicity data that is robust and transparent in the methodologies and provides ready access to the ecological toxicity test results for a variety of risk assessors, and researchers within the agency. So how does ECOTOX fit into this NAMS training program? The next generation of chemical safety evaluation will use new approach methods such as high throughput screening and computational modeling to connect and transform the traditional components of chemical risk assessment. ECOTOX provides data for the hazard evaluation component with accessible, structured, empirical toxicity data, primarily from in vivo studies, which supports standard chemical risk assessments, identification of data gaps to guide targeted additional testing, development of computational models, and development, evaluation, and adoption of other new approach methodologies. Specifically, ECOTOX conducts comprehensive literature searches and reviews of toxicity data in open and gray literature, such as government documents and reports, to identify and curate ecotoxicity test results, which are updated quarterly to the public website. We're showing the public website here and the address. Throughout its over 30-year history, ECOTOX has developed and refined transparent standard operating procedures that meet requirements for systematic review protocols. ECOTOX does not require logins to use the website, but through Google Analytics, we know that each month over 8,000 distinct hosts access ECOTOX. Users include researchers in academia, government agencies and contractors, state and local agencies, as well as the private sector. We are continuously reviewing documents and references for inclusion in ECOTOX. And so this quarterly update here, and these numbers change every quarter. The curated toxicity data in ECOTOX are used for multiple applications. Within the US EPA, records from ECOTOX are used to support environmental decision-making by program offices and regions. For example, data from ECOTOX are used in the Office of Pesticide Programs Effects Divisions for chemical registrations and re-registrations. ECOTOX is used as the source of citations and data for ambient water quality criteria documents. We have been working with the Office of Pollution Prevention and Toxics to be the source for ecological toxicity data to meet the objectives of the Frank-Lautenberg Chemical Safety for the 21st Century Act. ECOTOX is also used as a source of data for many ecological site assessments and in emergency responses by the Office of Land and Emergency Management. And over the last 3 years, we've conducted a major effort to provide ecological toxicity data on PFAS to researchers in the joint work group of the EPA Ecological Risk Assessment Forum, as well as the Department of Defense. So more generally, we know that ECOTOX data are used by federal, state, tribal, and international agencies for a variety of applications, including those listed here and described on the previous page for use within the EPA. We are also aware that data from ECOTOX are used as a component of many tools and applications, including the development of toxicity reference values and benchmarks, such as the ecological soil screening levels, species sensitivity distributions, such as the US EPA's interspecies correlation estimations, and NOAA's chemical aquatic fate and effects database. Records from ECOTOX are also used to calculate predicted no effect concentrations and ecological thresholds for toxicological concern, as well as in quantitative structure activity relationships and other modeling. And there are a variety of other applications there we are also aware of, but I can't fit them all on the screen here today. To support the integration of data streams used by many of these applications, we have been adding linkages from ECOTOX to other databases and resources, including the CompTox Chemicals Dashboard and other chemical-based databases. We're also working towards linking to species and pathway tools, applications, and sources, as well as reference management tools such as HERO. All right, so I want to give you an overview of how the data get into ECOTOX. This is a question that we get a lot, and so we'd like to provide an overview and then some details. So this is provided in the context not only of the ECOTOX specific steps in the pipeline, but also the general overview steps that are used in a lot of systematic review for those of you who are familiar with those processes. And so the general steps are in gray and the ECOTOX specific steps are in blue. So first, the pipeline starts with a chemical verification and development of search terms. We look for the information for chemicals that are requested These are typically come from program offices or regions or researchers. And then we develop search terms based on the chemical name, the CAS number, and other identifiers, as well as identifying synonyms along the way. We then run the literature searches through a variety of different search engines, including ProQuest, ScienceDirect, Dissertation Abstracts, Agricola or PubAg, a Web of Science, and we also have an internal database that we use. This is an example that we're showing from about a year and a half ago, where we did a large search for perfluorinated compounds. After we searched all of these databases, we ended up with about 37,000 citations that we downloaded. And then we removed the duplicates and we moved forward 8,651 for title and abstract screening. At the title and abstract level, we are looking for applicability to ECOTOX. Specifically, is it an ecological species that is exposed to a single chemical with a known duration, and those that don't meet the applicability get tagged for the reason why they aren't there, such as it is a chemical method study, it is a human health study, it's a survey, there's no toxicant in the study, it's a review paper, we only do primary literature, or it's a mixture paper. In this example, from those 8,653 references, 388 were forwarded for full text review using that same applicability criteria. And of those, data were fully extracted from the acceptable papers, of which there were 245 references. While ECOTOX does not include a formal study quality evaluation step within the tool, we provide a lot of information to help study quality evaluation assessments that are done for specific applications. I'll talk about that in a little bit more in a few slides. So this is the overview picture. I'm going to go into detail on each of these steps a little bit more as we go forward. So typically, a chemical is requested either by a program office or a research project. This is where the process starts. So once the chemical is requested, we verify the chemical abstract services registration number. We search various sources for chemical terms and synonyms and eliminate poor search terms. This can take a while, maybe an hour or so, but we have been working on developing a web-based tool to help identify and document these relevant search terms to not only improve the efficiency of this process, but also improve the transparency and documentation of where the search terms came from. So once we have developed a set of search terms based on the requested chemicals, we then take those and put them into this literature search engines that I mentioned previously. So now I have two different aspects on the top here. I have the chemical-based literature searches that I just described, where it starts off with a requested chemical from a program office or a researcher. But we also want to try to keep up as best we can with the current literature. This is not fully possible. We have identified 11 highly relevant journals from the history of curating data for ECOTOX. These 11 journals have high applicability for ECOTOX. So every month we look at the table of contents and review the titles and abstracts there as well. So there's two ways that papers can get into the pipeline for ECOTOX. As I mentioned before, we search multiple search engines, and currently this is being conducted with a semi-automated batch search through a tool that was developed by the EPA that's called Abstract Sifter Plus. You may be familiar with Abstract Sifter that is publicly available and searches PubMed, and this is a variation on that searches multiple literature search engines. So we send those search terms through the multiple search engines. We get a whole bunch of citation information back. We collate those citations and remove the duplicates. And then we look at them at the title and abstract level, which I'll talk about in a second. And each year, 100,000 to 400,000 references are screened for applicability. So it's a constant cycle. So from those literature search results, I said they move into the skimming for the titles and abstracts. And we use exclusion criteria that I will talk about in just a second to eliminate those that are not applicable to ECOTOX. This historically has been done manually, although we have recently incorporated filtering and artificial intelligence tools to improve this. And we are working not only with EPA offices, but other companies to modify their existing programs with ECOTOX specific strategies and criteria so that we can continue to denote the reason for exclusion and have some ECOTOX specific aspects of the AI tools. So those that make it past skimming titles and abstracts for applicability go forward to acquire the PDFs so we can look at the full text. And then when we review the full text, we're using that same criteria for applicability, but now we have access to a lot more information because we can look at more of the methods and the experimental design and the species information that may not have been available in the title and abstract.
Jennifer, I'm sorry to interrupt, but we had a couple of people ask if the list of 11 journals that you mentioned was available.
Not at this time, because it does shift a little bit. We reassess that on an annual basis. And so providing it as a list here would be disingenuous to the history of it. But as you can imagine, some that are typically very relevant are aquatic toxicology, environmental toxicology and chemistry journal. So there's a few that I'm sure the users are familiar with.
Okay, thank you.
Any other questions right now?
There's another question, but I think it's a broader question we can save till the end of the.
All right. So as we go through this process, I've mentioned it, I think, twice already, but every quarter we are updating toxicity data to the public website, and each year over 1,000, sometimes even over 2,000 references are added to that public website. So talking specifically about the applicability criteria that we use, this requires the species to be taxonomically verifiable. So we can verify the species and it's ecologically relevant. We don't curate data and look at data on bacteria or humans or monkeys. So this is really looking at wild organisms that can be found. And this really started off focused on North America, although has expanded quite a bit over the years. Additionally, the chemical needs to be a single verifiable chemical that can be linked to a CAS number, and the exposure amount needs to be quantified either as a concentration in the environment, and this can be either measured, analytically verified, or a nominal concentration. There also needs to be a known exposure duration, and we prioritize those studies that have control treatment. Additionally, there needs to be some biological effect that is measured, whether or not there is actually a effect of the toxin or not. There needs to be an effect that is measured, and the publication needs to be the primary source of the data and not a review, preferably in full text in English, although there have been some instances where it has been valuable for chemicals or species that are underrepresented to actually go through the translation process. So these applicability criteria can also be translated into a PICO statement for those who are familiar with systematic review, where your criteria are defined as far as population, the exposure, a comparator or control, and an outcome being present. So as I mentioned before, the reasons that papers are excluded from moving forward in the pipeline or considered non-applicable are tagged for the reason for rejection. This is really important because we can document why a paper was not moved forward for ECOTOX. And it's also really important because there are other disciplines that may be interested in some of these papers that we've identified through our literature searches. So we have routinely shared citation lists with human health folks, mixture, folks who are interested in mixtures, as well as some sediment or survey data is a common one that we share. So now we get to the fun part where we actually have to go through the data extraction process. And I just pulled up an example here to show you how you can have a single reference and you end up with multiple records in ECOTOX. So this example, we have a single reference that had a study on the exposure of tadpoles of African cloud frog Xenophus laevis to BDE 209. And in that study, they looked at multiple different effect group So looked at multiple different types of specific effect measurements that fit into different general effect groups. So the specific effect measurement, one that they measured was thyroid hormone receptor beta mRNA. So this was a change in transcription. And so that's the general effect group genetics. And in their study, they found a LOEC, or a lowest observed effect concentration. They also looked at some cell characteristics, so the height of the cells in the thyroid. They did some histological measurements in the thyroid also. They looked for residue in the organism. They also looked for development, specifically looking for the emergence of the forelimb. And they also looked at survival. So this is where you can go from one paper and one study with one species to having multiple records within ECOTOX, because each of these will have a record. So in addition to those toxicity test results that I showed on the previous page and are shown here in blue, where you have a results with an endpoint, an effect, and a specific effect measurement, we extract all the study details and methods in ECOTOX to cover a wide variety of different pieces of relevant information, including the unique identifiers for the chemical and the species, and a lot of information on this test species as shown in red here, specifically thinking about the species group it falls into, what was the life stage that was exposed and measured, what sexes were included in the study, where did that organism come from, what was the source, as well as information on the chemicals. Was there chemical analysis done? What was was it formulation? What was the grade of the chemical? What concentrations were measured, etc. And then as far as test conditions shown in green here, we have a wide variety of measurements to get at what was the exposure conditions, experimental design, etc. So you have, was it done in the lab or the field? What type of exposure type was it? What type of exposure media? How long was the study duration? How long was the organism exposed to that compound? And was there a control or not? So through the 30+ years of curating data, ECOTOX has established controlled vocabularies that cover most of these, especially those that are not values or categories. So the categorical fields all have controlled vocabularies, as shown in the asterisks here, and you can find those controlled vocabularies on the ECOTOX help page. It's called the terms appendix. So as I mentioned previously, we don't have a formal study quality evaluation component within ECOTOX as of yet, but many of the fields included in the data extraction can be used to inform study evaluations. And many of the criteria used in the evaluations are already required for publications in ECOTOX. For example, is the test substance identified? Well, yes, that is required for inclusion in ECOTOX. Is the species given? Well, yes, we have to have a verifiable species for inclusion in ECOTOX. Is there a control included? Yes, where control is a priority for inclusion in ECOTOX. In addition, a lot of the test conditions and test results are include fields that are used for study evaluation, such as what kind of test method was used, what was the experimental design, what were the water quality parameters, what was the statistical significance or significance level, and you keep going on to the whole list here. Got a longer list you can look at if you'd like to talk about study quality evaluations and how they overlap with ECOTOX fields. Please reach out to me. All right, so I'm going to pause there for any other questions at this moment before I move on.
Perfect timing. We have a couple of questions coming in. One of them was in response to a few slides ago, there was a question about journals. And then someone asks, there are databases, not journals, right? So is there a slide where you have a list of databases?
Oh, right here. Yeah, so these are the search engines that we use, and each of these search multiple different journals.
Great. The other question is, if an endpoint is not reported, is it included in the database? I guess the absence of an endpoint.
So I'm just going to go back here. So if there is a measurement for a change in something, so like they looked for a change in survival and there was no change in survival, we would still include that as evidence of no change. So even though there was no change in survival, that's still an important negative result from the exposure of that organism and the study. I think that's the question that's being asked.
Yeah, and everyone, if you find yourself wanting to clarify, feel free to just go ahead and do that in the chat. So there's a question about water accommodated fraction studies. Are those included?
I'm going to have to follow up on that question because I'm on the fly, not quite understanding it. So that sounds like a very detailed question, and I'd be happy to follow up with the person who asked it.
Okay. How is effect or endpoint severity considered? Are there mRNA levels for thyroid receptor model or considered as a different severity or potential impact versus frank mortality or developmental endpoints?
Right, so it's kind of the level of severity or the amount of change that's being documented by these test concentrations. And so that's going to really depend on what is reported by the authors, because what we capture in ECOTOX are author-reported effects. And so what I'm showing here are where authors reported low X or no X, or reported their data in such a way that we can interpret the results in low X or no X. If they reported an LC50 or an EC50, that would be included here. We also have additional fields where we can capture trend and statistical significance, which can help the user of ECOTOX interpret some of the severity questions. But really it's that interpretation piece is application specific. And so we really encourage all users to pull the data that's been extracted from ECOTOX. But when you get to those specific questions about study design or effect magnitude, that really needs to be going back to the primary literature to look at it.
Okay. A question about how is substance level data kept up to date? Like if new test data is generated.
So I think I'm capturing the question of how do we make sure what we have in ECOTOX includes everything in ECOTOXicology? And the answer to that is that we can't. So if the question is how do we keep all references included that are potentially relevant for ECOTOX, we can't. We do our best to review the new literature of those 11 relevant journals on a monthly basis and incorporate those that are applicable to ECOTOX and as we can, but we are limited by resources. If the question is instead specifically asking about keeping information about the chemicals that are represented in ECOTOX up to date as far as their CAS numbers and other information, we do routine updates for many of these fields, including the species data. So we update the species taxonomic information on an annual basis. We update the chemical information for CAS numbers, DTX SIDs from CompTox Chemicals Dashboard and DSS Tox on a routine basis. And then we update the effect groups, effect measurements, and other controlled vocabularies on an as needed or as identified basis.
Okay. Thanks, Jennifer. Just one really quick question, and there's a couple more, but I think we'll try to cover them later. So this one is just about whether co-exposures, so binary mixtures of two chemicals or multiple stressors, are excluded or included in the database.
Yeah, at this point, we are only capturing single chemical exposures. And so if a study includes a mixture, but then also includes each of those individual chemicals independently, we include the data for each individual single chemical exposure, but we are not curating or capturing the data for the mixture study. No, this is a major interest across many groups, and we have been struggling with coming up with a way that we can capture mixture data when really we need to be able to tie the exposure and the effect that are measured back to a single CAS number. So mixtures are not included at this point.
Okay, thank you. We'll keep recording your questions and try to come back to them. So keep putting them in the chat, and we'll let you move on with the next part of your presentation.
All right, so we are going to talk about just a few basic features of the ECOTOX user interface, which is where we put all the updated toxicity data, again, updated every quarter. And then I'm going to go through a demo of ECOTOX. So hopefully this will answer some of the questions. But I really appreciate the questions we just got in talking about some of the details on how the data get into ECOTOX. Those are really valuable questions, and I hope that I answered them. And if not, please follow up with me. All right, so some of the basic features for ECOTOX is that we have several different methods in this new user interface. It still feels new to me, even though it's a few years old. Well, you can either search or explore to start your query. And so most of the times you come to ECOTOX, you know exactly what you are looking for. You have very clear information, and you go to search and you either are overwhelmed with too much data or not enough. So search is really good if you know exactly what you want and you have these strong or hard parameters that you have to follow. For example, you need to have only a single species, no related species, only a single chemical, no related chemicals, and a specific exposure duration. Then you probably will want to start and search. However, oftentimes it is better to start and explore because you may know what you want, but you don't know how what you're looking for overlaps with available data in ECOTOX or available data in the ECOTOXicology literature in general. And starting in explore allows you to zoom in and out on the data available based on the parameters, and you can see on the fly what data might be available for your application. So to help develop a search, we have a tool that's in here that you're welcome to use, but the main point is that you need to do a little bit of planning before you start. Establish a process and a workflow, or just develop a plan, an approach to where you're going to go. So if we go into help, we do have a search planner. It's located under the select search parameters, and it's a PDF, and it has a form that helps you think about what chemicals do you want and what are the CAS numbers for those compounds, what species am I interested, am I willing to include related species, where you're looking at a whole genus or just an individual species. You can select the fields that you're interested in looking at, as well as other query parameters that you want to use to limit your search. So we recommend going to the search planning form to really think about what compounds you're looking for or where your starting point is, and whether you want to first explore the data in ECOTOX to have a better sense of what's available, or if you have really stringent criteria, or you're repeating a search that was done before, and then you can go straight to search.
Would you suggest not to separately search the databases and sources that the ECOTOX database searches? Or is the algorithm for the ECOTOX database internal search limited? So I think they're asking, would they duplicate themselves if they searched the ECOTOX KB and then also the sources that the database?
The sources that we use to identify the literature?
Right.
It depends on how recent we have conducted a search for the chemical of interest. And so if we conducted a search and have updated the data in ECOTOX in the last year, you may find a lot of duplication. As long as you're using the same inclusion criteria that we use for ECOTOX, you might find a lot of duplication in the references that you find. However, if your chemical of interest is one that we have not updated in ECOTOX in the last 5 or 10 years, then we highly recommend going out and looking for newer literature that wouldn't have been captured in the previous search because it's been published since we completed that.
Great, thank you.
All right, so before we get into this into the demo, I do want to talk a little bit about how you can go about looking for more information and helping yourself find what you need easily. So on the ECOTOX website, we do have a help page, and on the help page, we have a whole long list on the left-hand side, which is a table of contents that's got a lot of information. In this top section, you can see the criteria for including ECOTOX under this limitations link. You can download the ECOTOX user guide, which we update annually. Or you can scroll down on the left-hand side, you can see the list of field names and their definitions. You can find the terms appendix that I mentioned previously, and a lot of other information about related databases that you might be interested in. So lots of options on the help page. We also encourage people to contact us. And this contact us gives you a phone number or a website you can contact. And many times when people contact us, their answer to their question can actually be found in the frequently asked questions section. And so when we get questions through outreach or contact us, we add those regularly to frequently asked questions. So we do recommend, if you are waiting for an answer, you've contacted us and you're waiting for an answer, or you're feeling hesitant in reaching out, please review the frequently asked questions, because we do update those every quarter as well. All right, so now we are going to move on into our demonstration. So I'm going to start with a very quick demonstration of kind of a breeze through of every aspect on a very high level of the ECOTOX user interface. And then I'm going to go through a live demo a little bit more slowly so that you can see everything fast. And then when I go more slowly, we can stop for questions and answers as I go. So in this first demonstration, we are going to first start on the home page. And on the home page, we have the date that it was last updated, the stats and what's in the database. We have the chemicals that were most recently completed and updated in ECOTOX. We also have a variety of links to the terms appendix that I mentioned, the user guides, as well as some other links over here. So I'm going to just highlight these other links right now because they are important for some of the questions that just came up. And one of those is this recent additions. So in addition to what we have listed here is the most recent updated chemicals, you can go to recent additions and you can see the chemicals that have had significant changes during that quarter, as well as other changes that were made to the user interface during this quarter. And you can scroll down and see the last few years of recent updates. To go back to home, you can also go out to this literature search dates table, which is a work in progress. We're adding more information or expanding the information we'll provide here in the next 6 to 12 months. But for now, it's really valuable because at least you can see when a chemical search was last conducted. And you can type a name in here to search for a chemical name. You can see that we have these most recent ones also all have hyperlinks to the list of CAS numbers and that were used in those searches. So you can see this list here corresponds somewhat with the list here because sometimes the search, but isn't a one-for-one because sometimes the searches may have been conducted 6 months prior to when they show up on ECOTOX. So some projects are very large and we have lots of references that we need to go through and lots of data extract. All right, so now we're going to talk about the search option for querying information in ECOTOX. On this search page, this is similar to what the previous web-based query options were, but with a different kind of interface for the users. And here you can search, put in your search parameters by chemical, adding the CAS number or selecting a chemical group, adding specific effect measurements or effect groups, adding specific endpoints that you want to look at. These are the statistically derived endpoints that are reported in the paper. Searching for a specific species or species group. Limiting the results to specific test conditions that are part of your requirement or what you're looking for. And then also publication options where you can restrict the results to specific publication years, specific authors, or the specific ecoref numbers. So these are the unique numbers that are given to each reference that gets added into our backend. However, we're not going to go through this right now. You can see you have a lot of options for limiting your queries, and the challenge may be that you end up with too much or too little to reasonably sift through after you do this query. So I always recommend people start in Explore, where you can start exploring either by chemical, creating a custom group based on names or CAS numbers, or selecting a predefined chemical group. Start exploring by species, where you can create a custom group by species or select a species group. Or starting by effect, where you can create a custom group by effects again, or select a general effect group. So in this first quick example, I'm going to start with a species group. So I'm going to explore by species and then go into select amphibians and explore the data. So here on this group summary in Explore, I can see that there are 221 amphibian species with data in ECOTOX. And one thing of interest in this top left-hand corner, you can see these data include both aquatic records and terrestrial records. The ECOTOX database is actually still split into an aquatic set of data and terrestrial set. So when you query the data, you have to query these independently, but in Explore, you can see them side by side, which I find really handy. And for amphibians, it's really important because you have both life stages that are aquatic and life stages that are terrestrial, so they'll end up in both databases. On this group summary page, you can do sorting down in this table, or you can filter things. So for example, I could type in a genus here, and it would reduce my table just to that genus. This is lithobates, including leopard frogs. Or you can go on the left-hand side in the query filters, and if I scroll down a little bit more, you would see there's a spot to limit the genus to lithobates. And so then you would see my number on the top changed here to 16 species instead of just filtering the table here. There are a few options in Explore. You can look at records, which I almost never do because it's just a summary, and I'll show that in the next example. I like to go to the plot view because I'm a visual person. And so in this plot view, we have provided the results in a scatter plot and in a dot plot. And you'll notice here that there are 4,340 records, but not all of them are considered plottable. And what do we mean by plottable? Well, in order to put effect results together on a figure, we need to have them all have the same concentration units for the chemical exposure concentration. And so we consider records plottable if we can convert them to the standardized concentration units of parts per million or milligrams per liter. So for many aquatic studies, this is very easy to do. For many of the terrestrial studies, it is a little bit more challenging, so we don't have standardized concentrations for all of the terrestrial studies. And we do for a good portion of the aquatic studies. So now let's just look at this plot. You can see this example here. This is actually a custom plot that I created previously, where on the y-axis, we have the concentration of exposure in that standardized parts per million on a log scale. And then on the x-axis, I had done the custom plot option to put the species down here and to put the general effect group as the legend, so the different color and shape. And each of these is a record in ECOTOX associated with a concentration, an effect group, an effect measurement, and an endpoint. So if I hover over one of these, I'll see that this one is a development effect that was looked at. It was actually specifically looking at stage, and you can see the species and the concentration. It's a very low concentration, and the endpoint that was reported here was a low EC, and I get that eco-ref number. So this is the lowest point in this set of data that could be converted to that standard concentrations. You can also click on that, and below the table there is a very simplified table of the results from ECOTOX. So this one highlighted is that point that we just looked at on the plot. And you can see it's got just 12 fields. We have a lot more fields than that in ECOTOX. There are 300 to 400 fields in the output if you select everything, and about 90 entities that are actually captured. So an entity would be a species, all the species information, all the effect information, all the test conditions, etc. So this is real simplified. So I don't recommend anybody download this table and use it as is, but it provides you a little snippet view of what's available in ECOTOX, as does this plot. And here you can also see the reference right away. So if you hover over the reference, you'll get the full citation, and you can also get that unique eco-ref number, which you may use in search if you choose. And then we've also provided a link to the Google Scholar based on the title. So if you have access to that journal, you're able to get the PDF right away. We've also included a link under the chemical name for, it's about 80% right now, but in a few weeks, it'll be 95% of the compounds have been mapped to the DSS tox substance ID. And so then you can use that link here to go out to the Comptox chemicals dashboard, where you can get a myriad of other information about the chemical on that line, including all of the properties, other hazard information, exposure information, bioactivity data from the in vitro screening, etc. All right, so we go back here. So we've done our search. We're going to go back out to the group summary and bring us back out to all of the species of amphibians. You can do a lot of other query filters on this left-hand side. I'm just going to do some chemical queries right here. I'm going to take these chemical groups and select 3 groups. You can select multiples over on these drop boxes, and then that will limit us to now we have 32 species of amphibians that have data for those 3 chemical groups. You'll see still that we include data from both the aquatic and terrestrial. And so when we send this to search, which is what we're going to do next, we have to pay attention to what we're actually querying. So we've got 32 species for 3 chemical groups, and we want to get more information. So we're going to send this to search. And when you read this little pop-up box, you'll see that selections from query filters will pre-populate in the aquatic search tab unless only terrestrial is selected as the habitat. So we know for this set of amphibian data that it includes both aquatic and terrestrial data. So it's going to pre-populate it in aquatic. If we also want to have the terrestrial data, we have to do that separately. I'm going to send this to search, and you can see that it, as the little pop-up box indicated, it pre-populates on the left-hand side your query parameters that you had in Explore. You have your groups and your amphibians, and you can update your search. And then these turn gray because they're updated. And you can see in the aquatic data set, there's 759 results. If we wanted to refine our query, we can click on any of these other ones. So let's say we're going to go to effect measurements and limit this to only being growth and mortality. And then you can see that turns green. And then again, we need to update our search. So now we only have 345 results. And let's go ahead and customize the output fields. Let me just go back 1 second. What's shown here in this table in search is the 20-ish, a little bit more than 20 common fields that most people are interested in looking at, including information about the chemical, information about the species, the concentration, the effect measurement, and that statistical endpoint, as well as the reference. However, you may want other information. So if you click Customize Output Fields, we have all of the other entities listed here that you can include. All of these are all the other fields. And so you can select them individually. We have a default that you can restore if you select some and then want to go back. We've also identified a set of study quality evaluation defaults that include things like controls, all the comment fields, information about water quality, etc. We'll just leave that here for now. And then the other piece that you want to know is what studies did this come from? So for these 345 results, we're going to click over to the references. It's this little tab here. And again, you can go to Google Scholar and get that paper if you have access. If we go back to the table, now we're going to export this data. So we can go export as, and we can export it in multiple different formats. The condensed format of Excel has the columns kind of collapsed where you have your operators and your units and your values for some of the fields in the same column, whereas the full will have all of the operators, units and values in separate fields, which makes it easier for calculations and sorting purposes for trying to sort on a value. You can also export it as a delimited file or as in a format that is easily imported into the EPA's SSD toolbox. So this is just a screenshot of what it looks like in the table. Of course, we're missing all of the other fields, but this is a snippet of what you'll see in the Excel spreadsheet. It is truly a spreadsheet. All right. You can also go to the down on the left-hand side. You can go and see what your applied parameters are by category. You can export this. This information is also included on your Excel exports now, and that is a more recent feature that we've added. And if you want to independently export information about the references, you can do that here. You can export as CSV or a RIS file. Again, these are the references that are associated with the records based on your query parameters. All right, so that was the really quick walk through everything, and we are just a few minutes past the time that we had hoped to be at, so I'm feeling pretty good. And I would like to walk through a live demo of a simple example. And as I get that ready, I can take any questions if there are any.
Yeah, I thought I would pose a couple of quick questions for you.
Great.
One of them is, are plants included?
Yes.
Great.
Aquatic plants and terrestrial plants are included.
Great. Is bacteria included?
No.
Okay.
We're not including bacteria.
Okay. And in the species selection, would there be overlap between Acris crepitans versus Acris sp?
So these are two different species indicators that we have. And so if it has been identified to the crepitans, then we extract it as that specific species. But sometimes studies are reported in a way that they can only be verified to the genus. And so those would end up in the Acris species. So there's not any overlap there in the records.
Okay. Is fipronil included in the chemicals?
Well, let's go see. We'll start our demo here. How would you go about going to look for that? You could, not quite sure what's going on with this annotation. Can you guys see the yellow line across the screen? Okay. All right, so we can, a few different things. We can look in the literature search dates and look for fipronil. And you can say, oh, well, I guess fipronil is in here. It actually was searched last for a complete search in March of 2016, and we can click on it right there. So this is an example where you go back to the question that was asked previously. Should I go back to the original sources that ECOTOX used? Well, if you're looking for anything that's been published more recent than 2016, yes, you probably should update that information. That doesn't mean that we don't have things more recent than 2016, but we actually don't. So there we go. So in our fipronil search that I just clicked on, you can see it actually included 7 chemicals. So it was fipronil plus some related compounds that were requested to be included with that search. If you wanted to just include fipronil, you could go here. And then you can see there are a total of almost 3,000 records, both terrestrial and aquatic data. And if we look at the plot, you can see that there's a variety of different effect types that have been measured. And I'm just going to keep using this as my example here. We go into custom, change our plot parameters. I'm going to leave the y-axis as concentration. I'm going to leave the x-axis as the effect group, but I'm going to change the legend category to species group so we can see what types of species groups have been data in for fipronil specifically. And so you can see here that there are a wide variety of different species groups. You may notice as I hover over these that they highlight in the table, in the figure, the plot. And if I click on it, it'll actually remove it from the plot. Then I can turn it back on. So there's not a lot of reptile data in ECOTOX in general, or for this chemical in particular. There is some bird data, primarily in the mortality category, some worm data, fungi, a little bit of algae data, flowers, trees, ferns, and shrubs, lots of mammalian data, a few points for mollusks, some points for amphibians, particularly in mortality, histology, and enzymes, lots of fish data across the realm of general effect types, lots of information for insects and spiders, as well as other invertebrates and crustaceans. All right, are there other questions I should address right now?
No, I think you can continue.
All right.
Actually, there's someone asking you to elaborate on the column called chemical analysis method and the difference between measured, unmeasured, and blank cells.
Okay, so I would recommend you go to the help and you go down to data field definitions and terms. And that was a chemical analysis field. So here's where you can get all of the definitions. Maybe it's in the test result. You can search this too. Oh, there it is. Okay, so you can find the term and its definition, which is really helpful. So analysis of quantitative analysis of water in the test chambers or field sites is considered a measured concentration. Concentrations that are not analyzed in test chambers or field sites are considered unmeasured or nominal. If you want to see the set of terms that are there, we've got a little pop-up here, but a better place to look for that information is in the terms appendix, which I'll show in a second here. Sure why it's going that way. Chemical analysis methods. So it's appendix P, and you can see each of the terms that are available in our controlled vocabulary for that field and the definitions. So oftentimes if it's blank, it means it hasn't been specified in the study. And so we can't fill in a field that hasn't been reported in the study. And that is a big challenge, not only in ecotoxicology, but across toxicology and science, that not all the information that we might want is reported in every published paper. There have been some guides that have been published recently on required or recommended reporting for ecotoxicology. There was a technical paper in the ET&C for that, and I know some of the other toxicology and human health groups are working on providing some guidance on recommended reporting information for publications.
Okay, thank you.
So I do want to spend a few minutes walking through another example if we have a pause in questions.
Yeah, go ahead.
All right, so we are going to go through an example, and in this example, it's a real life example in which I was looking at the aquatic life ambient water quality criteria document for selenium. And as I was reading it, I read a comment in it that said, there may be species more sensitive than the data available when this criteria was developed. And aquatic dependent wildlife, such as birds, were beyond the scope of the aquatic criterion derivation. And that got me thinking, well, I want to know how much data are available for selenium and birds that could be used if somebody needed to investigate that. It wasn't included in this document, but I'm really interested. So we could start that in search, and we know we're looking for selenium and birds. So we can go to the chemicals. We can go to the chemical groups. There is a chemical group down here for selenium, and update our search. You can see that we're in aquatic, but I want birds, which are going to be terrestrial. So I'm going to click over to terrestrial instead and change it to birds in that species group, update my search. So now I should be getting terrestrial results for birds, 1,655 results. And that's great. I can look at my references. There are 68 references, and perhaps that's all I need to know. I'm going to export this either as a delimited or an Excel and have fun. I don't need to investigate anything more. I'm going to do all of my post-processing outside of ECOTOX. That works great. And I'm guessing those of you who primarily start in search, this is exactly what you're doing. However, if this had come up with 6,000, we would have had a problem, because that's a lot more results to sift through. Or 10,000 over is challenging for us to easily deliver as a downloadable file. And so that you have to limit it further. So that's one of the reasons we recommend people start in Explore, where you can start by chemicals. So we know we're looking for selenium. And if we wanted to create a custom group, we could either have gone and looked up the CAS numbers we're interested in and enter them by CAS numbers here, or we could browse chemicals, you type in selenium, and you see, oh, there's a lot here. Maybe I need to go back to that planning form and figure out what form I want. There's a lot in here. Wow, okay, cancel that. I didn't know that from the previous step that I did. So I'm not going to do a custom group because I'm not sure what I want. But I think I'll go with the predefined selenium group that ECOTOX had used previously. So there's a selenium group. I can see that there's over 10,000 records from a bunch of publications from 1934 to 2020, and explore the data. All right, so now I know that this group consists of 35 chemicals. Again, more information than I had when I was just using search. Whether it ends up impacting your interpretation depends on where you end up going, but it was something I couldn't gather from what I did in search. I know that I want to look at birds, so I'm going to go over on the left-hand side and go to species group birds. Oh, of those 35, only 10 of them have data for birds. And you'll notice on this left-hand corner on the top, it automatically transferred me to just terrestrial. Whereas when I had done the amphibian example, still both of those were checked. So now we're just searching the terrestrial database for birds and selenium. I see that there are 10 chemicals. You can sort and query this. You can look at the records where you can see there's a total of 1,655 records, which should look familiar from what we just looked at. And of these, there are distinct records. So this gets people every time. So what's the difference? So here we just try to summarize provide a simple table that showed real brief overview. And so we have summarized these up to effect and endpoint. So you can see this is the chemical, species, its accumulation, and its residue. This is a chemical, the species, it's biochemistry, and its specific measurement and a NOEL. And so this is a one individual line here, whereas this one residue has 5 different records in ECOTOX. And that's why there's a difference between the distinct and total, because this residue measurement could have been conducted in multiple tissue types or for multiple different life stages. And so you end up with a different record for each of those. So I don't use that table much. I mostly just go straight to the plot view after I look at group summary. And I can see that although I said that terrestrial data is hard to transform into the standardized concentration units, we still get about 2/3 of these are plottable and show up on this interactive plot, which I can hover over. I can click on that. I can see it in the table below. I can scroll over to the right. I can again look at the reference and link out to Google Scholar if I wanted. I can further on this left-hand side, do a lot of other filtering. If I go down, let's say I just wanted to look at specific endpoints, so I only wanted to include those that had effects, not the no effects. So I'm going to include the LOELs and the lethality. And I can see what that looks like. And so, okay, now let's say I want to remove that filter I just added. I can go down to the bottom and reset it. It will reset us to our chemical group or our custom group that we created. So I'll have to change it back to just having birds. And now I'm just going to keep it birds and selenium group and send it to search. In this example, it's going to send it directly to terrestrial because that's all we have. I can update my search. I could, if I wanted, add additional restrictions or parameters for effects. Or let's say I just want to do specific test conditions. Let's say I just wanted to, I wanted to limit it to only those that had reported controls and measured chemical analysis, or only had were specific days of specific number of days at which the effect was observed at. So if you had you wanted to look for 2 to 4 day studies versus a 36-day study, then we do that down here. Or let's say you wanted to look for specific publication year. So this example, it was 2016 was the publication date. So maybe I want to include in here that I want everything from 2015 on, but maybe I don't. So then I'll just reset this little button here within each of these. You can also do a reset all, but I don't want to do that right now. You can also add the specific eco-ref number here if you just want to look at one paper, and we're good. And then we can export this. And I exported it as a full, and I'm going to just pull that up, and then this demo will be done. So I exported as full into Excel, and then all I did to this table was I made bold, and then I did little drop downs, and I just highlighted a few of the fields to help orient people. You can see the CAS numbers here are unformatted, which catch some people, but entry into ECOTOX, you can use formatted with the dashes or unformatted, but we exported in the unformatted form because Excel and some other tools like to turn things with dashes into dates, become problematic. And the chemical name, a bunch of information on chemical analysis, purity. Again, I just highlighted a few fields you might want to be looking for, the species names, organism life stage. And then this concentration 1, author or standardized, is the concentration associated with the effect that's being reported. So in this record here, we have a concentration of 20, and then over a few other fields, there is the units, so 20 micrograms per gram wet weight in diet is associated with, keep scrolling over, again, we provide all the information here, is associated with a growth effect, which specifically was weight and is a LOEC. So the LOEC in this study was 20. And if you keep scrolling all the way over to the right, you can see that this response site, of course, growth effect, weight would be a whole organism, but there are other response sites, so that's part of the body. The observed duration, so that's the day that the measurement was taken. And then the units for that, so you can see that's days. And then the citation information at the end, including the eco reference number that could be valuable if you want to look it up again in ECOTOX. All right, so let us get back to, sorry, that was much quicker than I was intended, but I do want us to move into the case study phase where you get some hands-on experience.
Jennifer, as you say, there's we need to move into the breakout groups, but there was maybe let me ask you. There are a couple people asking about the SSD toolbox and whether it's possible to demonstrate that. Is that something that can be done fairly briefly, or?
That I think would need a whole different piece. I'm happy to share with folks what I know, but it won't fit into today's training.
Okay, so maybe that can be a follow-up to,
yes,
to be sort of a suggestion to include in future trainings.
Yes.
And there may be a couple of quick clarifying questions while this poll is going. So it's about the literature search date. So when this person searches the literature search date for chlorpyrifos, the listed date is November 2014. But then when they search chlorpyrifos in the search function, they find articles from 2015 onward. So
yes.
Can you explain?
And so that, yes, the literature search table is when the last comprehensive search was done for a chemical. So that's the last time we took that chemical and put it through the literature search process through those different search engines. However, if that compound shows up in any of the recent studies that we identify through looking at the table of contents and looking at those titles and abstracts from the recent publications that we do on a monthly basis, we will include that data in ECOTOX.
Okay.
That's a great question. Thank you.
Yeah, I thought that might be useful. Maybe another quick question, is the ECOTOX data cited in EPA CompTox the same data used in ECOTOX Knowledgebase?
There is a subset of the ECOTOX Knowledgebase data that are included in the ToxVal ECOTOX records. However, if you're looking for the broader set of ECOTOX records, we recommend that you come to ECOTOX because what's in ToxVal is a subset at this point, not only of the number of records, but also you don't get all of the supporting metadata and the additional fields. We are working with the ToxVal folks that have that ECOTOX table available in the dashboard. We're working with them to figure out the best route to include a link based on the reference, so you can go easily from the CompTox Chemicals dashboard ECOTOX table into finding that information in ECOTOX without having to try to recreate some queries.
Okay, great.
And then I'm just going to drop in the chat, if I can do this while I'm talking, the link for information about the SSD toolbox. And so that's an EPA developed tool, but I am not the owner or the developer of that tool. We just have worked with them to create an export that would be easily imported into it.
Great.
We are ready to move into our case study breakout groups. And so these breakout groups are assigned based on your familiarity with ECOTOX, as you reported when you registered, and each of you have been assigned a breakout group, and those was included in the email that you got yesterday. It's a big table with your name somewhere in that list. And in these breakout groups, you will work independently on the case study worksheets with a moderator available to help and answer questions. And so the goal is to have everyone use ECOTOX on your own to query it, to walk through these questions in the case studies, but have somebody that can answer questions and also talk to the other people in your breakout group to see if you are running into the same problems. Hello, everyone. I think we're all making it back to the main session. Apologies for the shortness of these breakout rooms. It's an interesting first time experience for me doing that in a virtual training environment. We had hoped to have more time in those rooms to work with people, but there were so many great questions in the main session that we didn't want to skip over those questions. So I am going to share my screen again in just a second. All right, so we do have just a few minutes here. I'm hoping that everyone has made it back. That we got a whole bunch of folks back, and hope that your work independently in ECOTOX led you to some interesting discoveries and maybe working through some challenges with the moderators or other participants. And so is there anybody in the level 1 who wants to share any insight on how it went, what they learned from the process, or what challenges they encountered and whether or not they solved them?
So, hi, I'm Jocelyn Piero from the EPA. So, ECOTOX, I do human health, so it's not actually applicable to what I do, but maybe in the future when I have a different position, it will be. And then I ended up liking the Explorer more than I liked the search, because even though search is supposed to be more specific, I had a harder time getting it to narrow down to the specific cast number that I had. For some reason, the search was giving me like broader number of examples than the Explorer was. So I'm not really sure why that was, but the Explorer ended up being easier and I was able to whittle it down to specific things a lot. And then the challenges I encountered were that the page would not scroll left to right. Our person said that it's supposed to be able to, but I didn't have a bar or anything to do that. And so I had to make sure to have a screen big enough to enlarge the page so that I could see all of the options that were available. So I think that's a quirk that should maybe be worked on for ECOTOX.
Yeah, thank you so much for those comments, insight, and suggestions. The scrolling side to side is an issue we encounter that some people can solve by pushing the middle of their mouse down and scrolling, but in some instances it is problematic. And so if you can describe exactly what was challenging for you, send it to our support. We'll put it into the list to be fixed, and that's how it happens. You find something that's not working well, we want to hear about it. You find a bug, we want to hear about it. You find an error in a data record in ECOTOX, we want to hear about it and fix it as soon as possible. So thank you very much. And that challenge with the cast number is a new one for me, but I agree that whittling down to exactly what you're looking for in Explorer is much easier than in search. Search is very constrictive to being very specific. Okay, I think we've got time for one other comment or group comment on level 1.
Hi, my name is Bernadette, and I work for the Pesticide Bureau here in New Mexico Department of Agriculture. So I found it very interesting, and I chose the chemical atrazine. And I started out with search, so atrazine wasn't listed there, so I went back to Explorer. And then it wasn't there again, so I went back to search, then I went back to Explore, then I kept going, then finally I found custom group. So that's how I was able to find the chemical atrazine. And then I chose the mammal species group, and I came up with 15 results. And then I got terrestrial as which one to choose. And then the types of effects that were absorbed were accumulation, avoidance behavior, and reproduction for that specific species group. And the lowest concentration for the effects were genetics and behavior. And then it produced 2 papers. And I referenced the EcoRef. Then from there, it kind of got, the directions were, it got confusing because I was trying to look for update search button, but it kept, there was a search button, but it was green and it was on the right side. And then that's
Awesome.
where the breakout ended. So I
All right,
found
so
it
we need
very
more
relevant.
time.
Mhm.
All right. Thank you for sharing that. It's really great to have those details. And I see a whole lot of questions in the chat that we won't be able to address them all here, but there are some good points. And clearly it's the first time we've gone through this case study example. We do have some pieces that maybe we need to update and add more clarity. And I want to give a moment or two for the any level 2 folks to share their experience or any comments. If there's someone in level 2 who'd be interested in sharing. I know that the group that I was in only got partway through the worksheet.
Well, I'm used to working in search, and I really like the plot view function that you showed. I hadn't seen that before. So while I was in search, I was looking for plot view, and it was mentioned that, oh, you can only do that through Explorer. And then I tried starting from Explorer all over again, and I find I just like search better. I mean, it'd be great if the plot view could be integrated into the search as well as the Explore function.
Yeah, that's a great feedback. We've tried to figure out how to balance the 2 options because some people really do like starting in search, and that's what they're comfortable with, and that's what they're familiar working in. I'll bring that up to the developers and see what we might be able to do.
That's just a good way of looking at it. I mean, instead of trying to scroll all the way across, back and forth across your screen, it's nice to see it on your screen as a visual. Like you, I tend to be visual, and I really like that plot view.
Yeah, and I see a lot of other good comments coming in the chat, and we'll take these and mine them for suggestions for the both issues that are problematic for users as well as suggestions for future development. Got time for maybe one more level 2, and then we're going to have to do our summary and wrap up.
Jennifer, this is Naveen.
Hello.
So I have a question.
Yeah.
Am I audible? Am I audible?
Yes, I can hear you.
Yeah. So my question is, when I look into the ECOTOX knowledge base for one of the ingredient BEET. So when I look into the references, it is coming from Google Scholar. So it is only relying on Google Scholar, the data which you are taking, or you are taking from ECHA also and other database?
The link to the PDF, we found that, or the link to the reference, we found that the easiest way to provide everyone a way that they could possibly find the PDF was to send it to Google Scholar. So we're just sending the title to Google Scholar to then see if you have access and permissions to access the full PDF. That's just a link out for those records that are in ECOTOX. The search process for getting the data into ECOTOX searches 6 literature search engines. And so we're not using Google Scholar as one of those. That's just the easiest way to provide the most users with a route to access the full PDF if they need it.
Okay. Yeah. Thank you.
All right. I'm just going to move on because we are just 5 minutes shy of our ending time. And I do want to provide a little bit of a summary. And so ECOTOX has long provided curated toxicity data, and it's been used in a broad range of applications in environmental research and risk assessment. And we are going to continue to develop ECOTOX and include additional updates and maybe even incorporate some of the suggestions for the user interface that are coming from this training. But as we do that, we are going to maintain our comprehensive and quality review of the toxicity data, although we are also trying to increase efficiencies as we go. So we will continue to do this and provide the curated data on the public website. I just want to reiterate that it is available for use, but remind folks that we don't have all ecotoxicological literature published in all times included. There's just no way we can incorporate everything. We are able to conduct searches for 40 to 90 chemicals per year. Those are those targeted searches, as well as those monthly look at the most relevant journals for looking for recent publications on chemicals. So we'll continue to provide it out on the public website, epa.gov/ecotox, for use by the agency, as well as other state, federal, tribal, and international agencies, as well as the industry, private sector, academia, and anyone else who I may have missed in that list of categories. But I really appreciated working through this process. This is our first round on this, and definitely have some great ideas of how to improve the case study breakout groups. And I appreciate all of you being guinea pigs for that. I see there's still some questions coming in and feedback on the comments, so we'll improve those as we go. And I invite you to reach out to us. I also want to thank not only the whole group of participants here, but the ECOTOXicology Knowledgebase team, which includes Dale Hoff and Jason Berninger at the lab that I'm at in Duluth, the Great Lakes Toxicology and Ecology Lab, as well as some folks at SCDCD, Colleen Ellenin and Sarah Vliet, the contract staff, both GDIT and SBS, and our senior environmental employment staff that all support ECOTOX and make it possible. And then we had a wide range of volunteers that helped us with these breakout room. And I really appreciate all of these moderators stepping up and making it possible so that we would have smaller breakout groups for this large group of training today. So thank you all, participants and contributors. And then I also want to highlight that as Jessica said at the beginning, there are future NAMS trainings being planned, and there's some potential topics listed here. As you can see in the ecotoxicology category, this is the ECOTOX Knowledgebase training, although there was enough interest that we would potentially consider future trainings as well. And if you want more information on the NAMS training program or future trainings, you can see the link on the bottom. And it's my understanding that the recording of this training will also be available on that NAMS training website once it has gone through its processing. And so in conclusion, I'm going to put my contact information up here. As I said, please reach out to me with questions, concerns, or comments. You can also reach out and send an email to the ECOTOX support. We also do have a phone number that is manned part-time at this point, since we are still somewhat remote. So ECOTOX support at epa.gov is the best way to get a response quickly. And if you want more information about this or the future NAMS trainings, please contact Jessica Daniel. I also want to highlight at the very end that although ECOTOX has been in development for decades, there had never been a single peer-reviewed publication that described the whole process. So as you can see through this set of training slides, I had referenced it a few times, but we recently had an overview manuscript published about ECOTOX that went through a lot of the details that I shared with you today.
Thanks everyone for joining.